Our next speaker is Professor Soumya Raychaudhuri. I'm not sure I'm pronouncing his name correctly. He is the MD PhD from Stanford University and currently a professor and also a doctor in rheumatology at Brigham Young and Women's Hospital, and also he is researching in human genetics at the Broad Institute. His main research topic is the how to understanding the genetic basis of rheumatoid arthritis and other immune-mediated diseases. Today he will give us a talk on molecular QTL in T cells to determine autoimmune disease mechanism. Please welcome Professor Raychaudhuri with big applause. Thank you.
Thank you. Thank you. Can you see my slides okay?
Yes.
Yes. Okay, very good. My lab, we think a lot about autoimmune diseases, and autoimmune diseases are a set of conditions where your immune system, usually your adaptive immune system, attacks an organ. And some of these can be very organ specific, such as multiple sclerosis. Others can be very non-organ specific, such as systemic lupus erythematosus. One disease that we spent a lot of time thinking about is rheumatoid arthritis. Rheumatoid arthritis was first described in the year 1800, or year 8 of the French Republic, actually in a medical student dissertation by Landry Bouveau. At that time, he had named it asthenic gout. His study had a total of 9 cases, which, by today's standards is unbelievable. And what he noted was that these individuals were predominantly women. They had an arthritis that had a chronic course and was polyarticular. And he made the distinction that this was different from gout, which is the well-known arthritic condition of the time. For a long time, it's been suspected that rheumatoid arthritis has a familial basis in studies going back, decades. The Scandinavian registries have given the clearest evidence of this. So rheumatoid arthritis is a disease that's present in about 0.5% of individuals. It's increased about affected monozygotic or dizygotic studies. As a result, our genome-wide association studies. As a result of the familial basis of rheumatoid arthritis, our genome-wide association studies have convincingly shown that RA has a genetic basis. We now have over 100 risk alleles that are associated with RA. Multiple sclerosis is another autoimmune disease. And as you can see, again, it also has a strong familial basis. If you have an affected parent, an affected sibling, an affected monozygotic twin, your risk of the disease goes up dramatically. And similarly, genome-wide association studies have identified large numbers of risk alleles, again, over 100 in these recent large studies, as you can see, of 50,000 cases, 70,000 controls. So what have we learned from these studies? Well, we've learned that T-cell biology is strongly implicated in most autoimmune diseases. The HLA locus is always the strongest association. It's intrinsic to presentation of antigen to the T-cell receptor and leads to T-cell activation. But outside of the HLA, there's a very interesting story as well. So 90% of the alleles are non-coding alleles, and only 10% are coding alleles. The non-coding alleles are disproportionately close to T-cell genes and are in regulatory genes that are T-cell specific. a possible mechanism that I think Keith alluded to in his talk and that we all think about is that you have a transcription factor. It would normally bind to a promoter in a very efficient way, but if there's a SNP there, well, maybe it binds a little bit less efficiently, and that has implications for how effectively the gene is expressed. So starting back in 2013 or so, we started using histone marks to identify regions of the genome that had that had enhancer activity, and we started to ask, well, which cell types are RA risk alleles most enriched in? And we found that indeed CD4 T cells is where we saw the greatest enrichment. Similar study by Kyle Farr and Alex Marson also showed that autoimmune diseases predominantly have their alleles in regions of the genome with CD4 T cell regulatory activity, kind of pointing in the same direction. So what would you expect then? Then you'd expect that these SNPs in regulatory regions would alter expression of a gene. You'd expect that things are expression quantitative trait loci. And so on the right, you see an example of this. So depending on whether you have a GG allele or a CC allele, the expression of this gene BLK, which is an RA risk allele, is higher, middle, or lower. And you see that in resting T cells in blue and activated T cells in red on the right. And this is a really nice example because here the risk allele co-localizes with the disease. So RS4840565 is the RA risk SNP in red. And here we've plotted all the SNPs with respect to their association with BLK expression. Strong expression with association. Okay, it's a strong EQTL, and when you condition on that, you still the SNP effect and RS are identical to each other. But most risk alleles do not alter gene expression. Many risk alleles, over 50%, are within T cell regulatory regions, but only 20% or fewer EQTLs co-localize with T cell EQTLs. So what are all those other risk alleles doing? And let me take you through an example. This is the CD28 CTLA4 locus. This is a SNP, RS117016553. And what you can see here in the bottom is when you look at that SNP in Jurkat cell lines, which is a T cell line, it has specific binding in an EMSA assay, suggesting that there's a specific nuclear extract that's able to bind the C allele and not the A allele. Consistent with that idea, on the right, when you do a luciferase assay to assess enhancer activity, you see that the A allele has some enhancer activity more than the PLG3 control, but the C allele has even more enhancer activity. Again, suggesting that this allele is indeed functional. That SNP sits in a regulatory region close to CD28, but you can see it has a direct connection with the CTLA4 enhancer in high C data. So all this points to the possibility that the SNP is regulating CTLA4 somehow, but the SNP is not an EQTL. When you actually look at actual expression data, you don't see any association. So what's going on here? So one possibility is that functional state might be the key thing. So is it possible that the SNP effect on gene expression is state dependent? T cells can take on many different functional states, of course. You can have a naive T cell, a memory T cell. Once you have a T cell that's transitioned from naive to memory, it can move on to become a central memory T cell or an effector memory T cells. They're very specific effector populations, Th17, Th1, Th2. And certain regulatory regions may be active within certain T cell states and less active in others. So understanding how these alleles work requires us to start thinking about which cell states they work in and what genes they act on. And this is essential if we are going to understand disease mechanism and develop targets for drugs. Again, a very simple schematic here. So you have an antigen presenting cell that takes an autoantigen. of some sort, it presents it to a CD4 T cell. That CD4 T cell is now transformed from a naive to memory T cell, and it can take on the right, any of these multiple effector T cell states. So we wanted to understand, do changes in T cell state, specifically T cell activation, ex vivo, influence gene regulation? So this was a proof of principle experiment done by Maria Gutierrez Arcelis, who now has an independent lab at Harvard, and Yuri Baglienko, a postdoc in my group. And for this, we did a simple experiment. We recruited 24 individuals, we stimulated their cells, and then we applied RNA-seq across a whole bunch of different time points. And then we asked, were there genes where we see evidence of allele-specific expression? And does that allele-specific expression change with stimulation? If we observed a change that suggests that gene regulation is changing with changes in T cell state. And then, of course, we would assess if these genes are disproportionately disease-related. So again, the proof of principle here was we had 24 individuals. We stimulated our T cells with CD3, CD28 beads. And we did RNA-seq at 0, 2, 4, 8, 12, and so on hours. And what we were looking for was evidence of allele-specific expression. So here at the bottom, you see that you have a gene, allele A and allele B, and maybe allele A has the C allele in the gene itself. So if you looked at those reads, you'd see more C than T, because you've got allele-specific expression favoring C. But now as you go through stimulation, the T allele is favored, and those reads are increasing while the C alleles aren't. So that's what we were looking for. So here's an example. Here's UBASH3A. You can see that the gene starts out highly expressed. This is an RA risk allele and a risk allele for multiple other autoimmune diseases. You can see that its expression goes down and then goes back up as you go out to 72 hours. And based on the presence or absence of a SNP, we can identify whether the transcript came from a reference or alternate allele. And now this is very interesting. So in the beginning, you see a lot of orange and not that much blue, but then the blue sort of catches up in the middle and then goes back down. And you can calculate a fraction. And now what you see is that in the initial hours, in the initial part of the you see that it drifts back towards 108 when we looked around 100. We found 186 genes when we looked genome-wide. My internet is probably a little bit unstable. Okay, so we found many examples of genes where we saw allele-specific expression. On the right, you see CXCR5. This is another example where over time, the reference fraction changes, that one allele is favored over the other. And on the left, you see that we had 186 genes. Many of them are of immunological significance. In particular, HLA-DQP1 shows a tremendous amount of allele-specific expression. And indeed, when we looked at these genes, we found that they were disproportionately enriched for autoimmune diseases. In fact, if you look at genes that were EQTLs for naive T cells versus those that had allele-specific expression versus those that had dynamic allele-specific expression, those that have dynamic allele-specific expression are dramatically enriched. So in summary, ex vivo activation demonstrated altered gene regulation in T cells, but ex vivo activation states may not represent what is occurring in actual tissue inflammation. So we needed to interrogate T cell gene regulation in vivo. This means that doing what we did in this last study of sorting cells, stimulating them, et cetera, may not be the best way to interrogate the genetics of T cell gene regulation, because we're looking at sort of artificial aphysiologic situations. So the solution that we decided to undertake was mapping EQTLs at single cell resolution in T cells taken in vivo and taking state into consideration. And of course, single cell technologies have just exploded over the last decade and have really given us the opportunity to do cohort level studies. What many individuals in the field are now doing is generating these single cell data sets, but we're analyzing them as if they're bulk data. So what is commonly done is single cell data is taken, is clustered, and then a pseudobulk analysis is done, which of course has limited resolution, forces you to consider discrete states rather than continuous states, which are so important for T cell activation states. Our goal here was to be able to map EQTLs at single cell resolution. And we wanted to find situations where maybe there was no EQTL if you looked at a lot of T cells, but in a small group, you started to see a bigger EQTL effect. And this is work done by Aparna Nathan, published earlier this year. How do we understand context-specific T cell gene regulation? So to do this in practice, we profiled memory T cells from 250 individuals. It was a Peruvian cohort where we assayed 500,000 memory T cells. We obtained RNA and surface marker data. How do you interpret RNA and surface marker data jointly? Well, we used an approach called canonical correlation analysis, and that really gave us the chance to take RNA and protein data and really put it in one space. So for our study, we had many surface markers for T cells. And what we did with CCA was we looked for linear combinations of genes and combinations of proteins that had a high degree of correlation with each other. And this method is a very old method. It's described by Harold Hotelling in 1936, but has become recently very popular in the single cell era. So applying this approach, we are able to see many different T cell clusters, as indicated here. We have 31 clusters. You can see there's a division between CD4 and CD8 T cells, CD8 being here on the lower right. We can see that there's a CD62L positive central memory population. We can see there's CD25 positive regulatory T cells. But these were clusters based on those canonical correlation variables that I just told you about. But those variables themselves pick up on those T cell states. So CV1, for example, picks up on cytotoxicity or innateness. CV2 picks up on the T regulatory state. And we did pathway analyses to prove this. So how do we model gene expression and how do we find EQTLs? So this will hopefully give you a flavor of this. Here we imagine that you have a log expression value, a certain number of read counts that you're looking at. You have a genotype main effect as indicated here. We model this with Poisson mixed effects model because the reads are so few and rare and sparse. Then we introduce a cell state variable. We have that main genotype EQTL effect. And now we say, well, what about differential expression for different cell states? What if you have a more cytotoxic T cell or CD4 T cell or CD8 T cell? That cell state variable can be a cluster membership, it can be an RNA principal component, or a multimodal canonical component. And then what we do is we add an interaction term, which takes into account the possibility that the genotype is interacting with cell state. Since every cell has a unique cell state variable, we can define a genotype effect for each cell state. So we can actually come up with an effect of the SNP on expression on an individual cell basis. So consider the map EQTL here. So here you can see this effect of the SNP is very strong in certain areas and nearly absent in others. And in fact, you can see if you look at the lower third of the cytotoxicity cells on the far left versus the middle versus the right, you can see that the effect of the SNP on expression is becoming larger and larger. The standard models that people use are linear models rather than Poisson mixed effects model we use. Apologies for this. I'm sorry. I don't know why my internet is being unstable. Can everybody hear me? Okay, I will go ahead and continue. So about a third of them, the effect of the SNP changed over cell states. One very interesting gene is MDGA1, which shows you how interesting and complicated these things can be. The same cell state may have opposing effects on two SNPs affecting the expression of the same gene. So here we have one SNP, which is the lead SNP. We have a second independent effect here. And if you look at the two SNPs, for one of them, the EQTL is getting bigger as the cells become more cytotoxic. For the other, it's getting smaller as the cells become more cytotoxic. And this is an example. This is ORMDL3. This is an autoimmune disease effect SNP. And what you can see here is that the SNP has almost no effect on expression for Tregs, a dramatic effect on expression for Th17 and cytotoxic T cells. And CTLA4, the SNP we started by talking about in the beginning, what you see actually has a very low effect in Tregs. And that's where CTLA4 has the highest expression. And it's actually in the cells where CTLA4 expression is low that the SNP has the biggest effect. So this is a very interesting example because for many cell state specific effects, the largest genetic effect is in lowly expressed cell states. So our analysis so far is focused on T cell EQTLs that are dynamic and genes that are EQTLs in low expression states may not be identified as T cell EQTLs at all because they may be averaged out. So the sort of genetic model tells you that it might be disease might be not just about cell states where the gene is highly expressed, but cell states where the gene is lowly expressed. And indeed, 71% of RA EQTLs are dynamic, and the same story is true for many immune diseases. And in general, EQTLs, if you look at them, are enriched for promoters. So dramatic enrichment for state interacting and non-state interacting. But if you look at those EQTLs that map to enhancers, that's where you see the difference. So the EQTLs that are most dynamic, they tend to be the ones that are enriched in the enhancer regions versus the non-interacting ones. So in the future directions, we are very interested in starting to look in tissue. All the work I've presented here is blood EQTLs. We've demonstrated how T cell EQTLs are dynamic, but are these EQTLs, are there EQTLs that are only seen in very specific cell states? The answer is yes. And we're working on trying to make our computational pipelines faster so that we can do more queries. And we're thinking about functional validation using SNP editing. So finally, to summarize, a single cell EQTL study reveals a broad range of T cell states. These T cell states can be modeled with continuous variables, which allow us to define single cell resolution EQTLs. About a third of EQTLs are dynamic across ex vivo cell states. Dynamic EQTLs are in fact enriched in enhancer regions. Many are disease alleles. Curiously, some EQTLs have their largest of genetic effects in low expression cell states. As we define causal genes in the context in which they cause disease, it is essential to think about the SNP effects and the gene expression independently. And I summarize my future directions. We're very interested in thinking about T cells in tissue, and we're working on doing that analysis because we think tissue is more representative, especially disease-relevant tissue is more representative of what's going on in disease. Finally, I'll just like to thank my colleagues and collaborators, especially Aparna Nathan, Yuri Baglienko, and Maria Gutierrez Arcelis, who led these studies, and a host of other people, in particular Socios and Salud, who helped us with the patient recruitment here, Alcus Price, Megan Murray, and Branch Moody, and their lab members who helped us with this study. And with that, I'll go ahead and stop and take questions.
Yes, thank you for the presentation. And I'm sorry that we had the technical difficulties due to the internet connection during his talk. I'm sure there will be many questions, and I cannot see the question at this moment. I think it will come up in a minute or so. Let me just ask one brief question, and how on the your single cell work, you in your regression model, do you have enough data that you are not overfitting your single cell data? I see many single cell lacks gene expression for for specific genes, and still you could find the regression coefficient reliably. What do you think?
Yes, that's a great question. No, we don't think we're overfitting the model for a couple of reasons. I mean, one, we really take into account sparsity because we use a Poisson model, which is good at modeling sparsity. Now, if you did a standard linear model or used a standard EQTL model that people normally use, you do have a lot of problems with overfitting. So that's the first reason. Second reason is actually the compared to the number of cells we have, we actually don't have that many parameters. It's really important that you have random effects in there in the model. I didn't get into this, but that's why it's a Poisson mixed effects model. The random effect is what allows us to kind of account for inter-individual random effects and technical effects and such things. And that actually has the effect of really constraining the model and avoiding overfitting. But that's an excellent question, and of course a major concern with this approach.
Okay, let me read another question. A model that has been developed in this study constitutes of three components like genotype, expression for cell states, and interaction. Is it possible to generalize and apply this model to different or smaller EQTL plus single-cell RNA-seq data sets? I think it is related to my previous question.
Yeah. So this data set had 250 individuals. We've applied this successfully to data sets that are under 100. I think it works okay. I would be very cautious about applying this model to data sets that are 30 or fewer. So I think, where you get into trouble is if you have too few people who have a specific genotype. So, and those problems, of course, happen dramatically when your cohorts get too small.
Okay. The other question is, okay, just a moment. Have you ever tried the first method of allele-specific expression with perturbation and applied it to single cell using long-read single-cell sequencing? I think it requires much more technology. Single-cell long-read sequencing. Not
in this study. We have another study where we're looking at like HLA QTLs where we do have long reads. And it's a very interesting problem because HLA has so many mapping challenges, which are unique to that locus. But no, not here.
Okay, I do have one more question. You showed the CTLA4 gene is relevant in your model. Did you look into PD-L1 or PD, since it is the best regulator of T-cell activities in tumors? PD-1, PD-L1?
So I don't know that we saw an EQTL and PD-L1. I mean, it's possible that gene is very highly regulated and maybe constrained. I think now it is a great question though. I think now that we're able to, look at more SNPs and more effects and more subtle effects, we'll have to go back and revisit. But in this data set, I don't think we saw anything.
You use the blood sample it regulates the body level regulation of T cell activity. So CTLA4 might be the prime candidate, but if you see go into the tissue level, tissue and organ level, some other immune checkpoint blockades should be important, I guess. All right.
Yes. So for an RA synovial inflammation, you know, about a third of the T cells are PD1 expressing, are PD1 positive. So I think that's absolutely right. I think if you look at the tissue, we have a better shot at finding these kinds of things.
Okay. Thank you very much for your great presentation.